Hello everyone. So we will continue where we stopped last time, talking about how non-disjunction of the chromosomes prove that actually genes are linked. And remember from Thomas Morgan experiments on Drosophila melanogaster, he was the first one to map a gene to a chromosome. A gene rather than the sex determination gene that Walter Sawton already proved. So this gene, the white gene in Drosophila, determine the eye color, and just by tracing or looking at the how the trait is inherited from basically the fathers to the daughters, then the daughters to the sons, this a pattern of inheritance suggests that the gene is X-linked because the fathers usually inherit the X chromosome to their daughters, while the Y chromosome goes to their sons, while X chromosome in females are inherited to both daughters and sons. So basically, last time we looked at what happens when the chromosomes fail to disjoin, fail to segregate, and basically we are looking here to one of the progeny of non-disjunction earlier happened in the mothers. Now this female has two copies of the X chromosome with a Y mutation and a copy of the Y chromosome. So what will happen in meiosis in these organisms? So if this happens in a human, mostly it will cause infertility because those chromosomes they will fail to segregate, so no functional progeny will be produced. But sometimes in very small percentages, these chromosomes will segregate. But how they would align? In most probable scenario, the two X's will align together in the prophase 1, while the Y chromosome, because it does not have a lot of homologous sequence with the X chromosome, so they will fail to align, while these two X's, because they represent a full homologous chromosomes, they will align together. So what we will get after meiosis 1 and meiosis 2, that these chromosomes will split, leading to formation of XY eggs or oocytes and a Y chromosome 1. But the other scenario is that the X and Y align together in very rare scenarios, and this will produce another two types of gametes, the XX together and the Y chromosome. And if we get XY from a father, a normal male, so we will get different combination of products. So now we will talk about sex-linked traits in a human and Y-linked traits in a human. And remember that human genetics is difficult to study compared to other model genetics because we cannot do selective breeding in humans. The number of progenies are really small, and the generation time is really long. So what Morgan discovered, what Walter Sawton discovered, and other model organisms can also be applied to human. So Wilson followed the inheritance of a trait, a disease, it's called color blindness, in which the individual cannot recognize between red and green. You will see them the same color. So normal individuals, they will see red to green as in this picture, while the color blinded one, they will see red green as brown color. So Wilson followed the pattern of inheritance of color blindness, and he concluded that the mutation is in a gene that is actually X-linked. So think about how could Wilson determine that actually this gene, how could Wilson map color blindness gene to the X chromosome. Think about it in few seconds. So he actually found that the inheritance pattern, the way of segregation of this disease, is that when the grandfather is affected, none of his daughters will be affected because they will be heterozygous. And then the grandsons of the daughter of that father, half of them will be affected, the other half will be normal. So basically, by following the behavioral or the segregation pattern of this gene, he concluded that this gene is X-linked. Uh, Another important evidence that this gene is X-linked is the ratio of affected males to affected females. So we know that males, they have only one copy of X chromosomes. So we expect that for X-linked recessive traits that the percentages or the frequencies of affected individuals are in males are higher than females, which is actually true for color blindness. So usually in Europe, some countries in Europe, in where color blindness is really frequent, the percentage of affected males reaches about 7 to 8% of males are color blinded, while less than 1% of females, they are color blind. This supports that this gene is X-linked, and it also supports the idea that the mutation is recessive. So there are other uh, famous diseases that are also X-linked, and they were among the first diseases that they were genetically followed and mapped, and they found that they are actually X-linked. Is hemophilia, the bleeding disorder. Individuals with hemophilia has a problem in blood clotting. If they get injured, it takes more time for these individuals to stop the bleeding, to clot, rather than normal individuals. And if we again follow the inheritance pattern of in a pedigree like this one, we can conclude that this mutation is in a gene that is X-linked. And the famous families that they had hemophilia, actually, the family of Queen Victoria, the family of Queen Victoria, they ruled England and different countries in Europe. So Queen Victoria herself, she was a carrier, meaning that she had normal X chromosome, let's say H plus, and a mutant allele for hemophilia. And here Prince Albert, because the square is not filled, he was normal, so he has karyotype XY, then he had H plus, let's say. So because he's normal, all of his daughters, not daughters here, daughters like Louise, Victoria, Patrice, they will be what? They will be, they will inherit the normal H plus allele from their father. But these girls, they have two options, two the probability have to get either the wild type allele or the mutant allele. That's why this female uh, is homozygous for the wild type, as the pedigree suggests, while this one, Patrice, she's heterozygous. She has one copy of the wild type and another copy of the mutant. Remember that here, very important, that females, they cannot inherit the Y chromosome from their father, otherwise they will become a male. So they have to inherit the wild type copy in this case. And they have two options. One of them is to inherit the wild type copy, the other one is to inherit the mutant copy. So here in this scenario, all of the Queen Victoria progeny, they were normal. But remember that males, they will inherit the Y chromosome from the father, and they have 50% chance to be affected because they can inherit either the mutant allele or the wild type allele. But none of the males, they were affected. Oh, here one. Leopard. Leopard, he was a son of Queen Victoria, and he inherited the mutant copy of the gene. And then his sons, they were normal because Helen, his wife, she was homozygous wild type. And then the daughter here, she was carrier. That's why half of her sons, they were affected. So hemophilia, similar to color blindness, is X-linked recessive trait in which it's inherited from either a heterozygous mother to half of her male progeny. Then the male progeny will pass the mutant allele to their daughters, and then the daughters will pass the mutant allele to their sons, and half of the sons will be affected. So similarly to X-linked recessive trait, we can encounter, or we can follow diseases that are actually X-linked dominant, such as hypophosphatemia. And if we look at the pattern of this pedigree, the pattern of inheritance of this pedigree, we can see that the inheritance pattern is vertical. So we can see that the disease is found in many generations, similar to autosomal dominant disease. So in every generation, there are many individuals that they are affected. But the big difference and the main difference between autosomal dominant inheritance and X-linked dominant inheritance is that when a male is affected, all of his daughters will be affected, but none of his son progeny, they will be affected. And you know why? Because females inherit the X chromosome from their father, while males, they inherit the Y chromosome. And most of these genes are X-linked, so there are no counterpart copy. There are no homologous copies on the Y chromosome. That's why the males are protected while the females will be inherited. But males can be also affected when the mother is affected, and if the mother is heterozygous, half of her sons, they have the chance to be affected, while some males can be unaffected because they have the chance to inherit the normal copy from their mothers. So one phenomena that is related to difference of X chromosome number between males and females is called the dosage compensation, which means that the X chromosome dose has to be balanced or equalized between males and females, because males and females, they have different X chromosome number. The way it happens in a human is by inactivating one of the X chromosome and forming a bar body, which represent a condensed inactive X chromosome. We can uh, recognize it by staining using a chromosome stain or nuclear stain, and we see that bar body usually move to the periphery of the nucleus. This usually does not occur in males because males, they have only one copy of X chromosome. So the timing where X chromosome inactivation happens, so after fertilization, so if this represents a zygote, At this stage, both maternal copy and the paternal copy is active, and it remains active until around a stage when around 500 cells to 1,000 cells are formed. At this stage, we call it the establishment stage, in which uh, half number of cells they will inactivate the maternal X chromosome copy, while the other half will inactivate the paternal X chromosome copy. Once this establishment of X chromosome inactivation pattern occurs at this developmental stage, after that a memory will be set. So all this will remember which X chromosome is active, which X chromosome is inactive. So let's say in these cells which decided to inactivate the maternal X chromosome, the daughter cells they will have the same path in which they inactivated the maternal X chromosome. While these daughter cells, they came originally from a cell that inactivated the paternal one, they will also inactivate the paternal copy. So females actually they are mosaics for X chromosome, and I am talking here about heterozygous females that they have, let's say, wild type copy for a certain gene and a mutant copy for the same gene. So some of them they will express the wild type copy, the other cells will express the mutant copy. So basically, females are mosaics. And one gene that resides on X chromosome, in which we can see this patchiness or mosaicism, is a gene that is important for sweat gland development. So all these sites, which represented by normal body color, they will have a cells that inactivated the mutant copy of the gene, while the one in blue inactivated the wild type copy of the gene. That's why they don't have sweat glands. So females will have mosaicism in the expression of sweat glands in females, in female cells. And this reminds me to tell you about the history of the discovery of this X chromosome inactivation, and it happens in calico cats. Calico cats are the street cats that we see them. So if you see a cat with mosaic uh, mosaic black and orange color, these are cats that are heterozygous. So the cells that inactivating the orange allele, the X chromosome that carry the orange allele will be black, while the other cells that inactivate the black allele will be orange. That's why we see a mosaicism or patchiness of black, orange color development in cats. The last thing to talk about is sex-influenced traits and sex-limited traits. So sex-influenced traits are traits controlled by genes that do not reside on X chromosome. They are autosomal genes, but the expression of these genes is different between males and females. One famous and known example is male pattern baldness, SALA, and we can see males they are the spread of this trait more common in males rather than females, because actually difference in hormonal regulation. So homozygous males and heterozygous males are bald, while only homozygous females are bald. Heterozygous females usually are normal. The other thing is the sex limited traits. So these traits are only present in one gender but not the other. Like let's talk about example like mutation in genes that affect pregnancy, affect the blood pressure during a pregnancy, sugar level during pregnancy, because pregnancy is only limited to females, not in males. The phenotype of mutation in these genes will appear only in females. One mutation that only affect males in the Drosophila, it's called the stuck mutation. So it's affect mating, in which males they cannot get separated after they mate from the females. So we talked about X chromosome linked genes. We didn't talk about Y chromosome linked genes. The reason is that because Y chromosome is a poor gene chromosome, only limited number of genes reside on the Y chromosome. So remember, the SRY gene is a gene that determines maleness. The other types of genes that reside on the Y chromosome are actually genes that are important for fertility. So if mutation happens in those genes, the male become infertile. So mutations usually are not passed to next generation. So that's it for this chapter. I will see you next chapter, chapter 5, about linkage and gene mapping.